Just the method of administration is what poses more of a risk. This is an injectable into the patient's body. The product has to be formulated and filtered correctly and filled correctly as per our aseptic techniques.
Aseptic fill finish is taking the client's product, we're formulating it to their specifications, then we're sterilizing it by 0.2 filtration, and then dispensing it sterilely into a vial, syringe, or cartridge.
Fill finish is not just about filling a product into a container. It's a multi-step process that is highly monitored and controlled.
Depending on the type of product and what the container closures they fill into, the formulation process, the sensitivity of the material. From what your target is defined, we try to develop the filling process and the formulation process towards that goal. The batch record include specific materials required for a manufacturing process and detailed stepwise instructions for operators to follow, and it has defined information and parameter and controls operators have to follow or document. And in the end, these are documented evidence of the testing results that support the product's quality and readiness for release. Once this master batch record is approved, it can be issued and printed out as it becomes lot specific. For that specific batch, you have a batch record. When you run a GMP batch, we want the master batch record ready so we can issue a specific batch record for the run. We also want the MOC that is in the implementation. Typically from regulatory world, it's change control. Change control means the creation of something new for GMPs. So these two need to be in place. The third thing need to be in place would be materials. You want all the materials already released, make sure it meets the quality standard and the quantity is right before you can make the GMP batch. Number 4 would be the equipment. Anything required for that equipment to be used in the batch manufacturing, qualification, calibration need to be checked off.
Specific materials we need to use for each aseptic formulation and fill are specified in the bill of materials. Group of associates in the warehouse will pull those materials. We put them into what we call a cave. The transport cart is locked. When it's ready for it to go into manufacturing, we'll bring that to the manufacturing floor, and then we will stage the material into the clean room. Component prep team will take all the consumable single-use items that are in a formulation or fill. They will wash it and prepare for sterilization and perform that sterilization. The formulation team is responsible for formulating product to the client specification. Once that formulation is complete, there could be a series of in-process tests to make sure that the formulation is exactly where it should be within specification. The formulation team will do the 0.2 filtration or the sterile filtration into the container that will then be hooked to the isolator to be filled from. So the formulation process varies greatly from drug product to drug product. Could be different mixing times, there could be different mixing speeds, there could be different order of adding the chemicals. It's the one process that we have which varies greatly from client to client because it can make a huge difference on the quality of the product. We use what is known as an aseptic connector. We use CPC aseptic connectors. to connect the formulated bulk of the filling line by attaching the two portions of the connector, locking them in place, and then removing the sterile membrane. So we're able to make a sterile connection in a non-sterile environment. Filling occurs within an isolator, and an isolator is grade A area. It separates the aseptic process, the aseptic filling, from the environment. Isolators are sanitized through a VHP process. After that process is over, they're put into what we call production mode, which starts an overpressurization, a laminar flow, and then the operators are able to do the manipulations through glove ports. After aseptic filling operations are done, the next operation will be visual inspection. The visual inspection team visually inspects 100% of the vials that are manufactured. They're looking to make sure that the vial doesn't have any defects, that the crimp seal is on and is firmly sealing the vial, and they're looking for any particulate matter within the vial. The visual inspection process takes place in different light conditions. The product has to be swirled to give us the best opportunity to see any particulate matter if it happened to be in the vial. So after the 100% visual inspection is complete, members of our shop floor QA team do an AQL sampling based on lot size, and then perform the same visual inspection as an assurance that the visual inspection was effective.
The role of the microbiology group at BSM is really twofold. They ensure the environment and the water that we use in our manufacturing is suitable for use. This includes monitoring for viable and non-viable particulate. The microbiology group also does post-filling assessments of the isolators to ensure there's no microbiological recovery after filling operations. The other major aspect of testing that the microbiology department does is on the product. So besides environmental monitoring, they also assess products for endotoxin bioburden, and they do the sterility test. At a high level, we have to ensure identity, quality, strength, purity, and potency. As a CMO, BSM is not necessarily required to perform all the testing, and we could share that responsibility with a product sponsor or other laboratories that the sponsor employs.
For drug product manufacturing, we follow the FDA guidelines 21 CFR 210 and 211, which speaks to the entire process of drug product manufacturing, from the incoming API, how it should be tested for us to use it for manufacturing, our components, how those should be tested for release for manufacturing, formulation of the product as well, and filling, the requirements associated with that, as well as the rooms, the grade of the rooms that we should be performing these processes in. These are all outlined in the CFR.
21 CFR 211.2 responsibilities of quality unit. There should be a quality control unit that shall have the responsibility and authority to approve or reject all components, drug products, containers, closures, and process materials, packaging material, label, and drug products, and the authority to review production records to assure that no errors have occurred, or if errors have occurred, that they have been fully investigated. The quality control unit shall be responsible for approving or rejecting drug products manufactured, processed, packed, or held under contract by another company. Adequate laboratory facilities for the testing and approval or rejection of components, drug product containers, closures, packaging materials, and processed materials and drug products shall be available to the quality control unit. The quality control unit shall have the responsibility for approving or rejecting all procedures or specifications impacting on the identity, strength, quality, and purity of the drug product. Every manufacturer will have to have a quality control unit that has these responsibilities. Having both quality assurance and quality control groups is a standard practice in our industry. The quality control group has a responsibility to test, inspect, and observe materials, while the quality assurance group takes that data and has a responsibility for disposition.
EU, we follow Annex 1. They tend to be a little bit more stringent with certain rules. With the Annex 1 revision, their main concern is POPSIT and the contamination control strategy. Their concern is the design of the clean room in terms of the flow of personnel and equipment. Someone that's newly gowned going into the clean room is not passing someone that's coming out, that's just formulated a product, right? Because that's how you can be contaminated, gown to gown. It's the same thing with equipment, it's the same thing with, at the end of a fill, how you're passing out waste, just to ensure that there's no risk of contamination with any new items going into the clean room. So it's the design of your clean room and how it's classified and the personnel and equipment flow. For FDA and EU, the preferred method for manufacturing, for filling actually, is isolators, as opposed to the open RAB system that a lot of companies still have. In the EU Annex 1, which was recently revised, the main focus is POPSIT. POPSIT stands for Pre-Use Post-Sterilization Integrity Testing. So it's pretty much testing the filters once they have been installed. For the EU, their rationale for requiring it is just to ensure that the filter tested on the line is integral. It is tested at that point because there's no more handling. So it's installed, it's ready to go, it's tested, it shows that it's integral, and then you can filter the product. Each clinical phase will have different requirements and purposes. For phase 1, it's the safety of the patient, just to ensure that the product is safe. Phase 2 is still the safety, but the efficacy of the product. How effective is it in treating the disease that they're in clinical trials for. And phase 3 is now the standard that's set for this product. The efficacy is all outlined, and now we will do process validation in order to move it to commercialization.
The requirements for raw material release differ at different phases of drug investigation and development. The CFR does allow for reduced testing of materials used for phase 1 drug product manufacturing. These exemptions do not apply at later phase. For a method verification, that normally is done to establish that an existing method works with a particular material. Examples of this are microbiology tests, where we have an endotoxin method. We do inhibition enhancement as a verification that our method works with a material. This type of work is used at all phases. Like you will verify methods for phase 1 through phase 3 in commercial products. This differs for qualifications and validations. A qualification is when we have a method that it's not established and we need to verify it with a product. We use the term qualification to differentiate from a validation in that at earlier phases, you need to show a method's fit for purpose. Generally, that is specificity, accuracy, precision, and linearity. And if we're dealing with impurities, limit of detection and limit of quantitation. That would suffice for a qualification. For validation, we need all of those, and we also need robustness studies that show how subtle changes could impact results from the method. And a validation is what would be needed for later phase 3, and commercial work.
When it comes to manufacturing, you need to narrow down what are critical and what are not as critical before you manufacture a batch, because those quality attributes will define what the kind of manufacturing process you're going to use. So critical quality attributes in that regard will depend on what is the intended use, route of administration, and strengths, and such. The other aspect is there's a regulation requirement on quality attributes for different drugs. For syringes, for lyophilized powders, and for liquid in vial, for stereo product, for non-stereo product, there are certain USP-defined quality attributes you need to meet. So combined with these two, you can define critical quality attributes. So at the phase 1, when you first dose into human, you usually concern more about the safety aspect. All these need to be defined. It's required. However, in the clinical phase, it's where you can refine your quality attributes. You know what's potentially critical, but you probably don't know what the range, how critical it is. But based on the clinical trials results, you could refine it. Critical process parameter is what you try to identify to control your process, such that you can always manufacture the product meeting the critical quality attributes robustly, consistently. When you have more understanding of the process, the list of critical process parameters should go down, because the number of those parameters should go down. Take, for example, some small molecule API. When you're not familiar with the process, you may consider mixing speed as a critical process parameter at the beginning, because you don't know it, right? But after you gain more knowledge of the process, you would find that this API is actually very easy to dissolve. Then this mixing speed becomes less critical process parameter.
There are several ways to aseptically fill drug product, each with their advantages and drawbacks. Biosafety cabinets are a great solution to mitigating cross-contamination from a room to the interior space of the hood. But a biosafety cabinet does have an opening on the front. The likelihood of bringing in particles and/or contaminants is higher than in a closed system. Open clean rooms offer one advantage, that mobility is much easier, but some pretty serious drawbacks. Sterility assurance is very difficult to offer. And it's not a preferred or common anymore way of filling drug product. An open RABS approach, there is a separation between the operator and the potentially sterile space inside. There are oftentimes glove ports or a vehicle that the operator works through. It allows operators ease of movement. For example, open a door on the RABS to fix any problems that are transpiring, and that wouldn't be possible on a closed system like an isolator. But doing so introduces high risk to sterility. With a closed RABS, it's relatively closed. Because of the regulated pressure differential, air moves in a known way. So not only is there a physical separation that can't be breached during the filling of drug product, but we know exactly the movement of any potential particles or even just the air. Isolator technology in filling of drug products is, in my opinion, the best approach. Unlike closed RABS, isolators offer a sanitization cycle, oftentimes vaporized hydrogen peroxide or VHP. So in addition to preparing the isolator with sterile items or sterilized items, it also has a sanitizing cycle that offers further sterility assurance. One drawback of isolators is the restriction of movement that operators encounter, but in our hands, we've been able to work within that restriction and achieve good filling outcomes. Sterility assurance increases the more you separate an operator from the actual filling process or the point of filling and dispensing drug. So as you move from an open area to an open RABS and then a closed RABS, all the way to an isolator, you're removing operators more and more from the process. And sterility assurance increases throughout that. An inline filler is going to look like potentially a rotary table, which is just a big round disc that's spinning vials around and around. And then an outfeed conveyor that will move one at a time through some barriers, much like a strand of pearls, one after the other. The vials stack up and move through the filler. In contrast, an XY table is, imagine something the size of a sheet of paper that is able to move in both the X and the Y direction. So away from you, toward you, and to the right and to the left in the filling process. Items, vials, cartridges, syringes, or custom containers would be set onto that nest as it moves around. Inline filling can be a very fast solution for large lots or time-sensitive lots if a drug is not stable, for example. But in the case of vial processing, glass vials bumping on each other along a single file line can be a pretty serious challenge. An XY table approach can nest, say, 100 units at once, which keeps the vial, let's say, or the cartridge or syringe, keeps them separate in their nest, which protects them from bumping or any glass delamination on the outside or damage. It can be a little bit slower, but you have much more flexibility in the type of unit that can be mounted to your XY table. When we think of manual filling, we're thinking of a human moving a dispensing needle to a piece of glass, like a cartridge, syringe, or vial, dispensing the product, and then manually closing that container. There can be pieces of that program, as mentioned, a needle movement machine or a capper, that are semi-automated, but you cannot extract the human movements from the process, and so we call it manual. Manual filling is often appropriate when lot sizes are small in early phase investigations, and it's more cost effective to fill 500, 1,000, 2,000 units manually than it is to start up an automated line and spend an entire day dialing in that process for high throughput if the batch size is so small that it doesn't justify the investment of time, personnel, and overhead. Peristaltic and piston pumps are fairly common across fill-finish CMOs. Peristaltic pumps are ubiquitous and utilize rollers to pinch a piece of tube and push product along through the tube. A benefit of peristaltic pumps is that the fluid path, the product contact tubing, is fully disposable. And so sanitizing the peristaltic pump is a simpler process and can transpire routinely, meaning the same high-quality peristaltic pump can be utilized time and again. If for some reason a peristaltic pump isn't appropriate, shear sensitive product or a very viscous product, there are a number of reasons, a piston pump may be more appropriate. One major difference is that piston pumps are direct product contact. And so the way the piston pump typically works is there's a chamber and a piston. Product will fill into the chamber and the piston will go down and push it out as a dispense. These pumps can offer higher precision at very, very low dose, but they're also very speed sensitive, not just in the dispense, but in the suction phase and in the rotation phase. Many of them have to rotate on their long axis to move from suction to dispense. They're also quite costly because they're product contact. They are not reusable without a cleaning validation or a client-specific program. Low-loss filling at BSM is a time pressure system. A low-loss system pressurizes a vessel, low pressure, maybe 1 psi for example, or even less, and then has a valve on the end of the vessel that simply opens and closes. And so it is one moving part, and that one moving part can be designed according to the compatibility of the client. We oftentimes use a valve that is PTFE, which is fairly inert, and The vessel that we pressurize can be made of plastic or glass or stainless steel. And glass vessels, fairly standard offerings include jacketed glass vessels that can help maintain temperature during this process. The benefits of a time pressure system are that if you use a high quality pressure regulator and a high quality valve, you can be extraordinarily precise. Our low loss system was qualified down to 0.1 grams of dispense weight, or 100 microliters, and could have gone lower. Losses on a traditional pump primarily come from tubing. In the low loss system, because there's a valve at the bottom of the vessel, tubing length is under 4 inches total. And any tubing involved in formulation is blown down under the bubble point of filters, of course, such that the total holdup of the formulation and filling process is around 15 grams. Sometimes we have been able to achieve less. In contrast, on a peristaltic pump with around 4 feet of tubing total, you cannot do better than around 40 to 50 grams of loss.
There's so much that goes into finding the right CMO partner, but I'll offer a few words of advice.
Look for quality and capability first.
Look at their audit history by regulatory bodies. Have they been audited by the FDA? What were the results of the audits? Did they receive any FDA Form 483s? How were the responses? What were the final classification of the audit?
The next I would recommend to look at how responsive they are, how collaborative they are, and on the people side, because to ensure the quality, you need people.
Is it high on their list that this is the best quality product they need to make, and this is, shared amongst all employees, that they see this as the main goal?
With the good people, with the capability and the quality, history, and mindset, I think you have a good setup for success.
Aseptic filling is an involved process, and it can look simple when it's done well. But a lot of science goes into the engineering of the process and into the training of the operators to perform it well so that it looks simple.
To deliver a high quality product, you need attention to detail and a well-designed process.
If you do it right, you will ultimately have a product, a high quality product that will save patient lives.